Why should we remove the macromolecules? Because we need the DNA in a very pure form. So that is the reason. DNA is negatively charged because it has a phosphorus backbone, right? So this negatively charged DNA molecule will always, because we know that opposite charges attract. So small volume cultures cannot yield applicable quantities of proteins. Hello everyone, a warm welcome to today's session on chapter 11 of second PUC, that is Biotechnology, Principles and Processes. I'm Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the tools in genetic engineering, right? Especially wherein we learnt about the tools that are used in recombinant DNA technology, wherein we had finished studying with the restriction enzymes that are involved in recombinant DNA technology. And we also learnt about how to amplify the DNA fragments or how to detect the DNA fragments by the polymeride gel electrophoresis technique, and also we learnt about the cloning vectors. In today's session, we shall continue with the other tools that are involved in the recombinant DNA technology. So to begin with, let us study about the processes of recombinant DNA technology. So recombinant DNA technology involves several processes, wherein the first process that is there is the isolation of DNA. Because if we need to go in for recombinant DNA technology, we need DNA, right? So if we need DNA, where are DNA is usually present? It is present inside the nucleus. And where is the nucleus? It is in the cell, right? So therefore, first we need a cell. Where is the cell present? Cell is the constituents of different plants and animals and different organisms, right? So first we need to select the organism of our interest. We need to take out the cells. From the cell, we need to isolate the DNA. So once we isolate the DNA, what do we have in our hand now? We have the DNA. So that is the first step. Next is, after isolation of the DNA, the next step that is involved is fragmentation of DNA by restriction endonucleases. So this we had studied, also we had studied isolation, and also a bit of this we had studied in the previous session, wherein how different restriction enzymes were used. We had studied about the restriction endonucleases. We studied the HIND3, which was one of the best example for the restriction endonuclease enzyme, right? So here, fragmentation of DNA. What do you mean by fragmentation? So fragmentation means you're cutting the length, one stretch of DNA strand. You'll not be using it as such because you don't want the whole genes that are present on the DNA strand. You just want a specific gene that is there on the DNA sequence. So you need to cut it. So in order to cut it, that particular process of cutting the DNA into smaller bits so that you can utilize the ones that you want is called as fragmentation. So for that fragmentation, what is used here? The one particular enzyme, that is restriction enzyme, called as the restriction endonucleases enzyme. And next, after cutting of the DNA strand, we need to again isolate the desired DNA. Now you don't get confused. First itself we had finished isolation. Now why should we again isolate? That was different. That isolation, the first step was you isolate a complete stretch of the DNA strand from that particular cell. After that, you cut it into small bits. From that small bits, now all the other sequences also will be there in the small bits, right? From that, which particular sequence strand you want, that you need to remove, right? So that is why we go in for isolation of desired DNA fragment. So after isolating, now you have, say for example, you have isolated 5 fragments which is having the genes of your interest. So those 5 fragments are now in bits, right? So can you use them separately? You will have to send them into the organism separately by using vectors 5 times if you have to go in for the genetic modification of that particular organism. So what scientists do here is, instead of sending them 5 times using different vectors, what they do is, they'll try to ligate that particular fragment. So ligation means you're trying to join it, you're trying to paste it. So it's just like cutting and pasting, right? So here ligation of the DNA fragment into the vector. So all those DNA fragments will be made into a stretch, and that particular stretch of DNA, what does it contain now? It will contain only the genes that we want, that is of our interest. So that will be sent into a vector. Now what is this vector? So vector is a vehicle. So as I told you, for you to move from one place to another, you need a vehicle, you need a transport, right? Just like that, in this particular recombinant DNA technology also, to make the DNA, to move the DNA from one organism to another, you need a transport, and that transport or vehicle is provided by the vector. So through that vector, what the vector will do, it will transfer the recombinant DNA into a suitable host. So from here you would have isolated the DNA, you would have cut the DNA into small bits using restriction endonuclease enzyme, then you will ligate the DNA and you'll put it into the vector. Now that vector will carry this DNA into the host organism. So that is what is done. What is done here. Now, is one small fragment in the vector, is it enough? No, you need to make multiple copies of it. So what happens here is, now you need to culture that particular host cell in a medium. Say for example, so now you need to produce an antibiotic. So there is a gene that is capable of producing one particular protein or enzyme that is responsible to provide resistance against a particular bacteria. Now, so such small gene product, if you get, is it enough? No, you have to supply it to the entire population of the country, right? So that is the time when you need to make multiple copies of it. So that is the sixth step here, wherein you culture the host cells in a suitable nutrient medium on a large scale, and after you culture the host cells, you need to extract the host cells from that particular medium. So that is the last step in recombinant DNA technology, wherein extraction of the desired product is done at the end. So all this we will study in detail today. So previous session already we have learnt about restriction enzymes, where exactly the restriction enzymes will go and sit. Where will the restriction enzyme go and sit? Will it go and bind wherever it wants on the DNA strand? No, right? We had learned. Please go back to the previous session, because all this is like a story. It is a continuous process that actually happens in the recombinant DNA technology. So when you want to read this, you also have to go back to the previous session and have a look at it as well, because there you need to understand where does the restriction enzyme go and place itself, what is its function, what does it exactly do, and all that. So those restriction enzymes, we had learned that they go and sit at a palindromic sequence, right? So that particular sequence will go and sit, and after that all this process will take place. We shall study about each of these in detail one by one. So first step in recombinant DNA technology was, we need DNA. For that we need to get the DNA from another organism. So to get the DNA, what do we need to do? We need to isolate the DNA. So that is the first step. So isolation of the genetic material, that is the DNA. So here, to isolate also, we need to cut the DNA strand. So here, in order to cut the DNA with restriction enzymes, first the DNA has to be pure, right? Why it has to be pure? Because in that DNA strand, there will be some other macromolecules also present. So those macromolecules need to be removed, and only pure form of the DNA strand should be got. It should not have all the other cell components and all along with it. So therefore, first is, in order to cut the DNA with restriction enzymes, it has to be, the DNA has to be in the pure form. We should have only DNA that is being isolated. The DNA should not contain any other macromolecules. So that is why first purification has to be done. So how purification is done, we shall look into. We all know that the DNA is enclosed within the membrane, right? Within the membrane, which membrane? So plant cell means they'll have the cell membrane and they'll have the plasma membrane. Animal cell means they'll not have the, they'll have only one membrane, right? So here, first what has to be done? The cell has to be lysed. Lysis means to break. Remember, lysis means to break, ligate is to join. So here the cell has to be lysed, that is the cells have to be broken. So first, if the cells has to be broken means the cell membrane has to be broken, right? So first we need to break open the cell. Why we need to break open the cell? Because the DNA is present in the nucleus of the cell. Where is the nucleus present? Nucleus is present in the cytoplasm of the cell, inside the cell, right? So if we need to get to the DNA, first we need to break open the wall of the cell. So we have to break open the cell, and once the cell is broken, the DNA will get released. So that the DNA gets released, and when it gets released, it will contain other macromolecules such as RNA. It will contain a lot of proteins, polysaccharides and lipids as well. Of course, it will contain RNA also, because who knows, at that time the transcription translation process might be going on. But here we don't need an RNA. We just need the DNA strands, right? That is the double helical DNA strand we want. So all these components which are the macromolecules, they need to be removed. So how do we remove those macromolecules? Why should we remove the macromolecules? Because we need the DNA in a very, very pure form. So that is the reason. So this can be achieved by treating the cells, be it bacterial cells, plant cells or animal tissues, with an enzyme called as lysozyme. What is lysozyme? Lysozyme is an enzyme that will lyse the cell wall. So lysozyme is a particular enzyme. Say for example, you need to isolate the DNA strand from a bacterial cell. So can you use any of the enzymes to isolate the DNA strand? No. If it is bacteria, then you need to use only lysozyme. So when you treat the bacterial cell with lysozyme, the bacterial cell wall will get opened, it will rupture, releasing, and therefore it will be easy for you to remove the DNA strands. So for bacteria it is lysozyme, and plant means it is cellulase enzyme, because the plant cell wall is made up of cellulose. So to break the cellulose or to disintegrate the cellulose, we need the cellulase enzyme if you need to extract the DNA from a plant cell. If you need to extract the DNA from a fungal cell or a fungi, we need chitinase enzyme, because the wall of the fungi is made up of chitinase. So that is why we need chitinase enzyme. So what are we doing in order to isolate the DNA strand? We are treating, if it is a bacteria, we are treating the bacterial cell with lysozyme. If it is a plant, we are treating the plant cell with cellulase. If it is a fungi or fungus, we are treating it with chitinase, so that the cell walls will get ruptured and it will be easy for us to isolate the DNA. Now the DNA that we have isolated, is it in the pure form? No, it is not in the pure form. So how can we get it in the pure form? Why is it not in the pure form? Because it contains a lot of macromolecules like RNA, lipids, proteins and all that, right? So we need only DNA. All that other things we do not need. So how can we purify it? So especially RNA. How can we remove the RNA from the DNA? So the RNA can be removed by treatment with ribonuclease enzyme, and proteins can be removed by treatment with protease enzyme. So ribonuclease enzyme, if you treat the cell or if you treat the DNA strand or whatever you have isolated with ribonuclease, then the DNA strand will be free of RNA. If you next after that you need to treat it with protease enzyme. So therefore it will be free of protein. So that is how now you have removed RNA, you have removed protein. So therefore, and other molecules are also there like lipids and all that are there. How do you remove it? So other molecules can be removed by suitable treatments, and after that the purified DNA ultimately precipitates out. How can you precipitate out? So what you do is, to that DNA strand you start adding ethanol. Ethanol is a form of alcohol. So you start adding ethanol. If you can see here, they have added ethanol to the test tube. So when you add ethanol to the test tube, only the purified DNA will come out in the form of threads. So this can be seen as collection of fine threads in the suspension. So that is how it is used by, further it is treated or purified by adding chilled ethanol, that is cold ethanol. So this is how it is done. So what did we do here? First we treated the cells with different enzymes based on what type of cell we are using for isolating the DNA. And once the cell wall ruptured, we got the DNA. That DNA is not in the pure form. It contains a lot of macromolecules such as RNA, lipids, proteins, etc. So in order to remove that, we had to treat it with different enzymes. So to remove RNA, we used the ribonuclease enzyme. To remove the proteins, we used protease enzyme, and to remove other molecules, different purification techniques was followed. After that, the DNA strand that was left out, to that the cold ethanol will be added. So when cold ethanol gets added, only the DNA strand will get separated in the form of thread. So that particular process is called as spooling. So you can see here, this is the DNA strand that is there, and it is in the pure form. Now, did we isolate the DNA in the pure form? Okay, we have finished the first step, wherein now we have isolated the DNA in the pure form. Now, since we have the DNA in the pure form, next step is to cut the DNA into smaller fragments, so we can isolate that particular gene fragment having the gene of our interest. So, next step is in recombinant DNA technology, cutting of DNA at specific locations. So, for this cutting, what is used? The restriction enzymes are used. So, these restriction enzymes will be poured to the DNA that we have isolated, and it will be left for incubation. Incubation means at certain temperature it will be kept as such without any disturbance for certain period of time. So what happens here is those restriction enzymes will start to act on the long stretch of DNA, and wherever the palindromic sequence is there, there it will go. So for specific site, there will be a specific restriction enzyme. So there it will go and particular palindromic sequence, and there it will go and it will start to cut the DNA fragments. So that is what it is done using restriction enzymes. Now after that, how it is done in the previous session, we have already discussed in detail about it when talking about restriction endonuclease enzyme. So you can just have a look at the previous session for that. So after doing that, now those fragments, all the DNA fragments will be there. Now from that we need to select only that particular fragments which we need. So for that we need to isolate the DNA fragment of our interest, right? So for that we go in for a procedure called as Agarose Gel Electrophoresis. So Agarose Gel Electrophoresis, what is exactly done here? This also in the previous session we have studied about it. So have a look into it. So Agarose Gel Electrophoresis is used in order to check the progression of a restriction enzyme digestion, whether the restriction enzymes have digested the desired fragments or broken the DNA having the desired fragments or not. That will be found out, and the separation of those fragments will be done using the Agarose Gel Electrophoresis procedure. So here we all know that DNA is negatively charged because it has a phosphorus backbone, right? So this negatively charged DNA molecule will always, because we know that opposite charges attract, so it will always move towards the positive charge. So what they do is, so I've already explained this, so to just brush up a bit. So what they do is, so in the previous session you can just go and have a look into the session, so you'll understand it better there. So they'll just pour into a tray, plastic tray, they will pour agarose gel. So that gel, once it's solidified, from that gel they will cut small pieces and remove it like this. So it will form a well. So into that well, they load this particular sample, whatever we have, that sample will be loaded. So what that sample contains? It contains the DNA fragments. Now the DNA is positively charged, right? And here an electric current will be provided, that is a negative charge will be provided. So when an electric field is applied there, those DNA fragments which are positively charged, according to their size, it will start moving on the gel electrophoresis. Those DNA fragments which are longer, heavier will move to a shorter distance, and those which are lighter will move to a far distance. So that is how now these bits of DNA strands have got separated. So that is what is done here. So DNA is negatively charged molecule, and hence it moves towards the positive electrode, that is the anode. So that is how they get separated. So after that, now we have on the gel electrophoresis plate, we have the separated DNA fragments. From that, which fragment we need, that we need to isolate. Say for example, we would have isolated 5 fragments. Those 5 fragments, what is done with those 5 fragments is the next step here. So here, the process is repeated with the vector DNA as well, and next is the joining of DNA is done. So why joining of DNA is done? By using several processes. Why? Because the DNA are in very small fragments. So that is why those fragments has to be joined back. So therefore joining of DNA is done, and after having the cut source DNA as well as the vector DNA with a specific restriction enzyme, the cutout gene of interest from the source DNA and the cut vector space are mixed and ligase is added. What they do? They now even the vector DNA also has to be separated. So vector DNA, so the DNA of our interest from a particular organism will be isolated. It will be cut into small fragments. It will undergo gel electrophoresis, all that, wherein those fragments will get separated. The same procedure is done for the vector DNA as well. So now we have on the gel electrophoresis, we have 2 sets of gel electrophoresis. One that is used for separation of the DNA fragments that have been isolated from particular organism, and one that is used for separation of the DNA fragments that has been isolated from the vector. Now we have these DNA fragments. Now these DNA fragments and the vector's DNA fragments have to be joined. What they will do is they will bring them together and they will add an enzyme called as ligase enzyme. What is ligating? Ligating is to join. For ligating or to join the DNA fragments of the whole of the organism of our interest and the vector, we need a ligase enzyme. So this ligase, when it is added, it will prepare the recombinant DNA. Why is it called recombinant DNA? Because you are recombining the DNA fragment. So that is why once you ligate it, once the vector's DNA and the host DNA and all, sorry, not the host DNA, the vector's DNA and the DNA isolated from the organism of our interest, those fragments are joined, we'll have a, because we are recombining it, it is called a recombinant DNA. Now what do we have? We have a recombinant DNA. Next, what is done with that recombinant DNA? We shall look into. So that recombinant DNA should be amplified because only one copy of it will be present. Something goes wrong with the experiment. Can you do all this procedure again? It is time consuming. It is also a loss to the lab, right? Because cost of this particular procedure is very high. So that is why prior itself what they will do is whatever recombinant DNA has been got, a number of copies of that will be produced. So that is the next step here. So amplification of gene of our interest using PCR, that is polymerase chain reaction, will be done. So here amplification means amplifying it, increasing the number of it, increasing the copies of that particular ligated DNA or recombinant DNA. So in this reaction, multiple copies of the gene or the DNA of our interest is synthesized in the laboratory using 2 sets of primers. One that is small chemically synthesized oligonucleotide that is complementary to the regions of the DNA and the enzyme DNA polymerase. All this will be used. So how the copies are produced, we learnt in the daughter DNA strands, right? Molecular basis of inheritance, how the strands get separated and the daughter DNA strands get forms to it to produce copies of the DNA strand. The same procedure occurs here as well. So have a look into that particular chapter, molecular basis of inheritance. So here what happens? First we have now only one, that is recombinant DNA. That recombinant DNA, we need to make copies of it. So to make copies of it, what they do is they use some primers, because for that complementary strands need to get produced, right? So for that primers will be used. What type of primers? One of the commonly used primer is a small chemically synthesized oligonucleotide, and the one more primer that is used is, and they also use an enzyme, they use an enzyme, DNA polymerase enzyme. So these are the enzymes and the primer that is used. and the enzyme will extend the primer using the nucleotides provided in the reaction and the genomic DNA as the template. So that is what happens here. So here this particular enzyme will help in extension of the complementary DNA strand, because DNA polymerase means once the primer has to go and sit at that particular site and it has to start producing the complementary DNA strand, then if the nucleotides has to get added to that particular complementary DNA strand, then DNA polymerase enzyme is needed there. So that is why along with the primer, DNA polymerase enzyme is also used here. So that is how copies of it is produced. The same procedure which takes place in a body naturally, like during cell division, how copies of DNA strands enters into the daughter cells, right? So there we had studied in the molecular basis of inheritance chapter. That same procedure here, but it is done artificially in the lab using polymerase chain reaction machine, using a machine and using a primer and a enzyme. So that is how it is done. Now we have amplified it. So what happens in the polymerase chain reaction? We shall repeat it again using this diagram, wherein the region to be amplified, that is that particular region. So which region? The recombinant DNA. This is the recombinant DNA, right? Don't get confused. It is not the DNA that we had first isolated. It is the DNA that after all the procedure, that final recombinant DNA which we get, right, which is having only the genes of our interest, that DNA I am talking about. So it is a recombinant DNA. So in that particular recombinant DNA, we need to select a region or that portion that has to be amplified, or that has to produce multiple copies of itself. So once it is selected, we need to denature it. Denature means we need to break it, right? So if we need to separate the strands, denaturation is required. So for denaturation, one of the best steps that they follow is heat. They heat the DNA strand, because ultimately the DNA is nothing but it consists of proteins, right? So those are amino acids. So those proteins or amino acids, they get broken or they get altered only when we supply heat to it. So that is why this particular DNA strand, recombinant DNA strand, which has to be amplified, that will be subjected to high temperature or a suitable temperature. So when it is subjected to a heat or temperature, the DNA strand gets separated. Now to that DNA strand, what are we adding? We are adding primers. That is a oligonucleotide primer is added. Oligonucleotide primer is added. Now that particular process is called as annealing. Annealing means to join something. You're joining something. You're annealing. You're pasting something to the strand of DNA. So along with this primer, what do we also need to add? We need to add an enzyme called as DNA polymerase, and one of the best enzyme that they have used here is Taq polymerase enzyme. So why Taq polymerase enzyme or DNA polymerase enzyme is used? Because the nucleotide should get added, right? So that is why. So here you add the primer. To that primer, the nucleotides will get added like this. How the nucleotides get added? It gets added because during the process of annealing, along with the primer, we have added the DNA polymerase enzyme. So that is why. So after this particular process, next is you need to extend the strand, that is the complementary strand. So extension will be done. Extension, where this process will occur, it will be done in the polymerase chain reaction, which is a machine. In that machine it will be done. So that machine will run for about 30 cycles, so that the same copies of those strands will get produced, because same procedure will occur on all the strands and repeated copies of those strands will get produced. Now you have the amplified, that is approximately 1 billion times the DNA has been amplified once 30 cycles of polymerase chain reaction occurs. Now how many copies of recombinant DNA you have? 1 billion copies of recombinant DNA. So how much experiments you can conduct on it, right? Instead of, so if you don't do this procedure, what will happen? Again you have to go in for isolation and all that. So that is why any procedure, once you get a recombinant DNA, always we need to go in for polymerase chain reaction so that multiple copies of it can be produced. So whenever we want for our experiment, that particular copy will be available for the experiment. So that is the reason. So this is how recombinant DNA technology, in which amplification of the gene of our interest is done. So if the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately 1 billion copies. So for 30 cycles means 1 billion times you're amplifying the DNA. So therefore 1 billion copies of DNA is produced. And every time, if the copies have to be produced, a thermostable DNA polymerase enzyme will be used, and this particular DNA polymerase enzyme, that is the Taq polymerase, which we had taken the example of one of the DNA polymerase enzyme, Taq polymerase. It was isolated from the bacterium Thermus aquaticus. That is why it is called as Taq, meaning Thermus aquaticus. It was isolated from this particular bacteria. And this particular enzyme is again a protein. It should be thermostable, that is, it should be capable of withstanding the changing temperature. So that is why a particular thermostable DNA polymerase will be used. One best example is the Taq polymerase enzyme. And this enzyme, it will actually denature the double-stranded DNA, and it will amplify the fragments, and later the ligation of the fragments will take place, and further the cloning of these fragments will occur using a vector, or this fragment if desired can now be used to ligate with a vector for further cloning. So here it can be inserted into a vector also, and these fragments can be amplified, or it can be cloned, or otherwise it can be cloned in the polymerase chain reaction machine as well. So this is how the amplification of the gene of our interest is done here. So now we have multiple copies of the recombinant DNA which is having the gene of our interest. So next work is to insert this particular recombinant DNA into the host. So what did we do? We took it from one organism, we are inserting into the host. So all the process we have finished now. After all the process, what we have in our hand? We have multiple copies of the recombinant DNA. What is there in that recombinant DNA? All the genes of our interest is there. So that recombinant DNA, now we need to put it into the host. So what is done here? We should look into. So for that, we need a recipient cell, right? Recipient cell. Recipient means something which receives. So that will be a recipient. So which is the recipient here? The host is the recipient, because the host organism is receiving the DNA from another organism. So here therefore the recipient cells, we have to make them competent, or we have to make them suitable to receive the DNA from another organism. So therefore the recipient cells, after making them competent to receive the DNA, they will take up the DNA present in its surroundings. So if a recombinant DNA bearing gene for resistance to an antibiotic, say for example, which is resistant to an antibiotic ampicillin, if that such recombinant DNA is transferred into E. coli cells. Now what is this E. coli cells? It is the vector. Why should it be transferred to the vector? Because the vector will be the one which will carry the DNA from here to the host organism, right? So that is why. So the host cells become transformed into ampicillin resistant cells. Why did the host cell become transformed into ampicillin resistant cell? Because the recombinant DNA which we are using has the capability to show resistance towards ampicillin. So that is why this particular host also develops resistance against the ampicillin antibiotic. So that is how the genes, whatever genes we are supposed to get it expressed in the host, that will get expressed in the host organism once the DNA incorporates itself in the host organism. So after that, if we spread that, now what we need to do? We need to spread the cells. So here the cells have to be multiplied. So we need to spread the transformed cells on agar plate containing ampicillin. So I told you how to make them ampicillin resistant. Say for example, to make them ampicillin resistant, we have to take a plate. In that plate, we have to pour media, that is a Petri plate. To this Petri plate, which is containing a nutrient medium, we have to pour ampicillin. We have to put ampicillin. After putting ampicillin, what we need to do is, we need to take the bacteria, that is E. coli. Now in that E. coli, what is there? The DNA from that has been isolated, the recombinant DNA that has been isolated, that is there in the vector, right? Now that recombinant DNA has to be made into multiple copies and they have to be made ampicillin resistant. What is done here? That particular vector will be plated or it will be put in this particular medium which is containing ampicillin. So when it is put into medium containing ampicillin, because it is resistant to ampicillin, wherever the ampicillin is there, these bacteria will not grow. Other regions it will grow. So that will show that the ampicillin resistant genes in this case is called a selectable marker. So selectable marker, if you remember, in the previous session we had studied about a vector, right? That is, we had studied about Prebia 322, one of the vectors or a plasmid we had studied, which had different regions. In that one, apart from the origin of replication region, there was a region called as the selectable marker region, wherein either you can use any kind of antibiotic such as tetracycline or ampicillin. So here, if we use ampicillin, that particular bacteria or vector will be resistant to ampicillin. So that is how now we need to find out, has all the vectors, so when you put that vector in a nutrient medium, they will start to grow. That vector is nothing but a bacteria, right? Obviously they will grow, they will divide and multiply. So when number of those bacteria are produced, say for example, number of bacteria are produced, do you think all these bacteria when they multiply, all will have the recombinant DNA in it? No, some will not have. How can you find out which is having the recombinant DNA and which is not having the recombinant DNA? To find out that, what they do is, already in these vectors, what are their selectable markers are put, that is the ampicillin resistance. So, see whether they are resistant to ampicillin. To this media itself, they will add ampicillin and they will allow the growth of this vector. So, here wherever there is ampicillin, that surrounding these particular bacteria will not develop because they are showing resistance to, or they will start to develop because they are showing resistance to ampicillin. Ampicillin cannot do anything to it. So, aram se they will start to develop there or they will start to grow there. So, when they start to grow there, it is understood that particular colony contains the gene of our interest or that vector carries the gene of our interest. So, that is how only those vectors will be selected and the other ones which have not grown will be left apart. So, that is how it is done here. So, after that, now we need to obtain the foreign gene product. Now, we have multiple copies of the vectors, right? That is the E. coli, multiple copies we need to have. So, for that, after that, we need to obtain the foreign gene product. So, how to obtain the foreign gene product? Let's look into. So, when you insert a piece of alien DNA into a cloning vector, which is this alien DNA? It is the recombinant DNA that we have inserted, right? So, when the recombinant DNA is inserted into a cloning vector, which is a cloning vector here, it is the E. coli bacteria that is being used as a cloning vector. And then it is transferred into either a bacteria, all these are the host. We are transferring into the host. What are the different hosts? Either bacteria, plant or animal cell. Then the alien DNA gets multiplied in the bacteria, plant and animal cell also. Because why will they get multiplied? Obviously, these bacteria, they tend to grow inside the host as well. So, they get multiplied. So, when they get multiplied, what happens? The number of recombinant DNAs also gets multiplied. So, that is how the gene product or the foreign gene product gets multiplied in the host organism. And in almost all recombinant DNA technology, the ultimate aim of this particular recombinant DNA technology is to produce a desirable protein. So, as I told you, why to produce a desirable protein? What will be there in the recombinant DNA? All the genes of our interest. And those genes will be producing, after transcription translation process, it will be producing the protein of our interest, right? So, that is why the main aim of recombinant DNA technology is to produce the desirable genes or proteins of our interest. Hence, there is a need for the recombinant DNA to be expressed properly. And can all the vectors that have multiplied on the nutrient medium express the recombinant DNA? No. How can we find out whether they are expressing it or not? Learnt about it in the previous slide, right? Wherein we had put nutrient medium along with ampicillin and we grew that vector, and that vector was ampicillin resistant. So once it grew on the ampicillin media, we could find out that okay, all the vectors are having the recombinant DNA of our interest. So that is why, so only those will be selected. So obviously those will express themselves when they are put inside the host organism. So the foreign gene gets expressed under appropriate conditions in the host organism. And after having cloned the gene of our interest with optimized conditions and the expression of the target protein, one has to consider producing it on a large scale. Now, we have put it inside the host and that is capable of expressing itself in the host by producing the proteins of our interest. Now, that particular gene or that particular protein has to be manufactured on a large scale. How it is done? So, the cells that are carrying the cloned genes of our interest. Where is it carrying the cloned genes of our interest? In the host organism into which we have inserted the vector, right? So the cells harboring the cloned genes of our interest, they are grown on a small scale in the laboratory in a proper medium, and these cultures may be used for extracting the desired proteins. And then the purification of these cultures will be done by using different separation techniques. So here the cloned genes that are there, we need to increase the number of production in the in various industries for industrial purpose and all that. So how can we increase it? If we leave it in the host organism, will it get increased to our desired extent? No. So that is why those cells will be isolated from the host. Why will isolate the cell? Because those DNA will be carrying the gene of our interest. So it will be isolated and they will be cultured properly and multiple copies of it will be produced on a large scale, and the separation will be done by using various separation techniques. And these cells which have been isolated from the host, they can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while the fresh medium is added. So they use a culture system wherein the fresh medium is added and the media from which the cells are separated, that will be drained out. As it is drained out, again and again the fresh medium will get added there, and that the fresh medium is added from the other to maintain the cells in their physiologically most active log or exponential phase. So we need cells which are in the growth phase or the log phase or the exponential phase. So such cells will be collected, and this type of culturing method produces a large biomass of the genes of our interest, therefore leading to high production of the desired proteins that we need. So that is how obtaining the foreign gene product is done, by mass culturing the cells which have been isolated from the host organism. What cells? Those cells which are containing the recombinant DNA having the genes of our interest. So for this particular procedure, some bioreactors are used. How they are used exactly, we shall look into. So small volume cultures cannot yield applicable quantities of proteins. So one of the best example I can take of is insulin. How we can manufacture insulin on a larger scale, because we know that half of the population is suffering from insulin despite being any age, right? For such population, insulin medications has to be provided. So that insulin medication has to be provided in a large quantity. Can you manufacture it artificially? It can be manufactured artificially, but the source should come from a natural source, right? So for that, what they do is they amplify the gene that is responsible for the production of insulin. Now that will be in the host. So the host cells will be isolated and those cells will be multiplied on a large scale so that large copies of the genes which produce the insulin can be got, and from that the proteins can be extracted. So therefore large quantities of insulin proteins can be produced. So that is how it is done. So to produce large quantities, bioreactors are available wherein it having a capacity of about 100 to 1000 liters. So such bioreactors have been developed which can hold a capacity of the cultures, of the cell cultures of 100 to 1000 liters, and these bioreactors, they are nothing but vessels. So into these vessels, as you can see here, it's just like a vessel. So into this vessel actually the cultures will be put, the culture broth, as you can see it. It will be usually 100 to 1000 ml capacity, that is, or 100 to 1000 liters capacity it will be there. So such capacity, so up to 1000 liters of culture you can put there, and once you put the raw materials, these raw materials will be converted into specific products, individual enzymes, etc. using microbial, plant, animal, or human cells as well. So that is how it is cultured. So what do they do here? They will make a culture broth. What is there in the culture broth? In culture broth, the host cells are there. Host cells carrying the gene of our interest, it will be there. Now those host cells have to be multiplied on a larger scale. So they put the culture broth into this particular bioreactor, which is a vessel. So after putting the culture broth, what is done? So now in this particular vessel, the temperature will be adjusted. So temperature will be adjusted, then the acid or base will be adjusted, then steam will be provided for sterilization. So all this will be adjusted. So bioreactor will be in such a way that will be able to provide all the optimal conditions that are needed to produce the desired product of our interest, by such as what are the different optimal condition. Optimum temperature will be maintained, optimum pH will be maintained, optimum substrate will be maintained, optimum salts, vitamins, oxygen, everything will be maintained. Like how a premature baby is put in the incubator, right? So how will the incubator be? Just like a mother's womb. Whatever the mother's womb can provide to the baby, that the incubator will be providing to the baby. Just like that here also, the bioreactor will be designed in such a way that is capable of maintaining a lot of optimal conditions related to temperature, pH, substrate, vitamins, salts, oxygen, etc. And this will actually favor the increase in the production of the culture, or increasing the production of the number of cells. So here usually one of the common bioreactor that is used is a stirred tank bioreactor. The stirred tank bioreactor is usually cylindrical, as you can see here. It is cylindrical, or it might have a curved base, that is bottom might be curved. So if you can see here, it is cylindrical to look. Cylindrical means it is pipe-like. You have seen cookers, right? Pressure cookers, just like that it will be. But pressure cookers base will be usually flat, but this will have a curved base. So this will have a curved base. So it is cylindrical, having a curved base. And what is the capacity? It is about 100 to 1000 liter capacity. And because of the curved base and the cylindrical shape, and because of the presence of a stirrer, if you can see here, this is a stirred bioreactor. It will contain a stirrer. This stirrer will neatly mix the culture broth very carefully, so that optimum temperature and all that can circulate throughout the culture broth. So the stirrer facilitates even mixing and oxygen availability throughout the bioreactor. So the bioreactor is cylindrical in shape, and it has a base that is curved, and it is a capacity of 100 to 1000 liters. Into the bioreactor, the culture broth will be put, and once the switch is put on, the stirrer that is there inside the bioreactor, it will start to rotate. When it starts to rotate, it will start to mix the culture broth properly, so that oxygen can circulate equally throughout the culture broth. And also the air that is being trapped can be bubbled through the reactor. So for that, the stirrer is very, very important. So the bioreactor, apart from that, it has an agitator system, and it has an oxygen delivery system, and it has a foam control system. it has a temperature control system, and it has a pH control system, and also it has a sampling system. So all this will be there in the bioreactor. It has a pH controlling system, then it has a motor which will run the bioreactor, and it also has a gauge like thing that will actually tell us the temperature so that optimum temperature can be maintained. It has a foam breaker because when mixing is done, because of the supply of oxygen and the breakage of the air bubbles, foam gets formed. That foam should not hinder the culturing process. So therefore a foam breaker will be there, and it will also have a section wherein the sterile air can be pumped inside, that is pure air, and also a section wherein the steam, or a pipe wherein the steam can be provided for the sterilization. So all this will ensure that optimum condition is maintained inside the bioreactor, which is needed for the proper multiplication or the culturing of the cells. And after completion of the biosynthesis, the product has to be subjected to a series of processes before ready for marketing. That process is called as downstreaming. Now, a number of cultures have been produced. From that, proteins have been got. Those proteins need to be purified before they are marketed. So, for that, downstreaming is done. So, the process includes that particular process of downstreaming. It includes separation and purification. And these two processes are collectively or together called as downstream processing, and the product has to be formulated with suitable preservatives, because they are also proteins which may get denatured later on after long storage. So that is why suitable preservatives and all that need to be added before they are marketed. So that is how various medicines, antibiotics, all that are produced on a large scale using the recombinant DNA technology, and finally the bioreactors, and then the downstream processing. So such formulation has to undergo a lot of, so before marketing, once the downstreaming process is done, wherein purification, all that is done, the product, final protein product that we get, that cannot be just given to the market as such. It has to undergo a lot of clinical trials. After that clinical trials, and if they find out that the drug is safe for use or that protein is safe for use, then only it will be further marketed. And strict quality control testing for each of the product is also done, and the downstream processing and quality control testing, so downstream processing as well as quality control testing, they vary from product to product. So it has its own standards depending on the product. So all that, if all those standards are met, then only the product that is got is considered safe and suitable for usage, and therefore it will be marketed, So this is how the final end product we get from recombinant DNA technology. So especially for manufacturing medicines and all that, this particular recombinant DNA technology is one of the best suited methods. So these are the different steps that are followed in recombinant DNA technology. So it is a very, very vast process, but how much is needed for the PU levels? So much that is there in the NCERT textbook, so much has been explained. So if you need to understand few of the portions, you need to look into the previous sessions as well. So concentrate on those previous sessions when watching this video, so that you'll understand better. So I hope you understood this session, wherein we completely understood the 7 steps that were there in the recombinant DNA technology, right from isolating the DNA to getting the end product and the marketing of that particular end product. So with this, the chapter comes to an end. We shall meet again in the coming sessions with a new chapter. Thank you.